Okay, let's get started. We will update you with our electrophysiological characterization of the mouse models. So today we're going to talk about E200K. So a little bit of introduction, especially for the parents. Basically, neurons are the major type of cells in our brain, and probably 100 billion of them. And left picture is to show you like a mouse brain section with lots of neurons labeled by different fluorescent proteins. They're very pretty. And on the right-hand side is individual neurons. As you can see, it's a distinct cell type from many other cells. It's a small cell body. And many like longer processes. And you can see from this right-hand side, the details of this process has lots of buds like the spring tree, lots of new buds coming out. Actually, those are the locus for communicating with other neurons. As I mentioned here, neuron major as two parts. One is dendrites for receiving signal from another neuron, and axons, which talk to another neurons. So basically, my lab tried to study the communication between neurons because they are critical for our brain function. So actually, we can measure them quantitatively in a laboratory session, and I will show you is a brain structure called hippocampus, which is critical for learning and memory. Actually, synapse there is very simple. The cell is big. It becomes a prototype of synapse for us to study synapse transmission beyond studying the molecular basis of learning and memory. And as you can see here, this is a slice of hippocampus. And you can see this dark area, these are cell body layers. Many of homogeneous type of neurons in that area. The CA1 area is the pyramidal neurons, and the CA3 area is all pyramidal neurons. So it's very homogeneous. And there's many beautiful features for this preparation because these synapses come here making connection only once with this dendrites of the CA1 neurons. They make technically the recording interpretation data is much simpler. So this is just schematic of the same thing here. And how do we measure communication between neurons? There are many ways. The simplest way, the most straightforward way is this. We can stimulate these axons from CA3 cells to make it fire. And then a glutamate was released binding to CA1 neurons. Then you can record this signal here. So experimentally, this is the trace will look like. So this is a signal here. So for technical reasons, we measure the slope of this trace. So that's indicative of synaptic transmission or communication between neurons. So in this case, it's specific between CA3 and CA1 neurons. So this is one of the many ways of measuring communication between neurons. So you can make this, on top of this, you can make many fancy experiments. One of them is you can stimulate these CA3 neurons twice with a little bit time interval between them. Then you get two responses. So you can, the ratio between these two responses actually is sort of a proxy to determine whether there are change of glutamate release probability in the presynaptic terminals. So we got our mouse model from Dr. Stefan's group, and thanks, Ron. Appreciate your help in all the coordinations, lots of bureaucratic stuff between the two schools. But finally, we got it. But they won't release the mice to us from quarantine until like last week or something. But anyway, we rushed some experiments on it. So this is a straightforward experiment to determine synaptic transmission between the CA3, CA1 neurons between control and the K in mice. We call this E200K. So we choose E200K because we believe it will be easy breeding according to Ron, and that's the case actually. We started breeding in the quarantine. We cannot access it, but the Valerian people help us, doing the breeding and cutting the toes, and we do the genotyping. And once it's released, we can start doing experiments on it. On the other hand, E198K, another one which gives much severe symptoms in the patients, it's very hard to breed. So we didn't try to work on that one at the very beginning. So this is the data here. We don't see difference between these two groups of mice here. So basically, the straightforward interpretation based on this assay is probably no change in communication between neurons between the control and the wild type, control and the K in mice here. So this is this mouse model directly mimicking human conditions. One copy wild type, one copy this E200K variants. Then we performed another experiment. We give stimulate CA3 cells, give two stimulus with a different time interval in between, 10 milliseconds, 15, 25, 50. So this is called path-post ratio here, PPF studies. So look, for the wild-type neurons, as you can see, it's going up and coming down. So if there's no difference, these two curves should be completely overlap. But here we see some kind of trend of an increase of this curve for the E200K mice here. So at this stage, we only work on 3 mice for each group, 8 slices and 6 slices each. We do not see statistical significance, but the trend is very clear. So that's kind of promising for us. Then we perform a third test. So this one we give like a 14 Hz stimulation. You mean every second we give 14 stimulations on CA1, CA3 cells. Then we record the signals from CA1 neurons. So at the very beginning, you see this increase of signaling. Gradually, this signal is going to decrease. The reason is you stimulate the presynaptic neurons so quickly, so you're going to deplete the glutamate. They get released. Normally, glutamate is going to recycle back and repackaging. You get lots of vesicles. But if you stimulate in a fast way, the system cannot keep up with. So eventually, you're going to deplete the glutamate. The response kind of going down. So this is what we call a depleting assay. So this is another assay to assay short-term plasticity for the presynaptic terminals. And again, for this case, we see the difference in terms of trends between the two groups, but still no significance yet. But our experience tells us if we do more slices, more mice, we probably can see a difference. So the interpretation, if they are significant, interpretation between this set of experiments and last set of experiments will suggest that maybe the glutamate release for the E200K mice is decreased. So if that's the case, then why we do not see change in this assay here? Because this assay, if a glutamate release probability decreased, but we stimulate it, so we can probably bypass that mechanism. So there's possibilities. So in that case, it suggests this assay may not be the best. We still have to go to like more difficult assays to determine whether synaptic transmission is different between the two groups. So I think that's all the data we have so far, and we need to do more experiments and working with more mice, more slices. Hopefully, we can decrease the error bar, get significance. If that's the case, we're going to perform different type of assay to assess synaptic transmission. And also, we need to study synaptic plasticity. And if all pans out, and there's a difference, that's very promising, because at the very beginning, we didn't expect to see difference, because this is one of the mildest mutations. The E190K probably will give us the strongest differences. But this is a pleasant surprise. We hope this holds out. So I think that's our data here. So these are the people who did experiments. All the phase was done by Carl, a graduate student in the lab, and Zara is a technician. She's doing all the genotyping. And Cynthia and Daniel, they're gonna work on the biochemistry, biochemical aspect of the project. If you have any questions, we're happy to answer you. Thank you. We have E198K, and that one, the global knock-in. I mean, just one wild-type copy, one mutant copy. That one's breeding, according to Ron, is very hard to get. So we haven't even touched them yet. Especially, we do not have access to those mice when they're in quarantine. So you need to take special care. We couldn't do it. So that's why we only focus on E200K. But for E198K, there's a flux version, so we can try to breed with the different crylines. So that will overcome all the issues. So we're gonna try to do, but that will take like 2, 3, 4 months before we can get the mice for studies. Yeah. So at present, probably we're gonna work with the E200K, because number one, there's a reasonable chance we got significant difference, and then we can work on the biochemical signaling pathways. This is gonna be in the presynaptic terminals. It's a very specialized structure, so it's very different from postsynaptic neurons. So that will be, it's gonna keep us occupied for now. Yeah.